So, this is we're August 7th, our virtual meeting between Cancer Support Community in San Francisco Bay Area and NorCalCarciNET Community. And we are with Mark, is it Krailo? Making sure I get your last name pronounced correctly?
Correctly, that is it.
And you're a professor of of research population and public health science. You're my stat guy because now in 3 years at working at the Vail workshop, I've worked with you twice, which has been incredibly fantastic every time we've done this. And one of the reasons that I wanted to do this, and we tried to do this at the Vail workshop the first time we met each other, but it was so windy it would knock over my cameras, is that, as patients, we're always dealing with multidisciplinary team, and it's know your team. And there are some unique parts of our team, like our pathologist, who we never meet. But as I sat there with you in the Vail workshop and listened to how statistics and how working with stats actually changes the trials that go on and the drugs that get approved for our conditions, I realized that there's probably more than one, but there's certainly one area which most patients never get to talk to and never really get to understand. And that is the statistician who helps set up all these trials and really guides it. And I've been fortunate, in working at the NCI, working on task force and clinical trials to work with some statisticians, and that's always part of a clinical trial review. But working with you in what I'll call the MASH unit of clinical trial design of phase 1, phase 2, where we go through 7 or 10 different clinical trials, it really brought to attention that one, our oncologists who are part of our team don't necessarily all know the stats that are behind their trials. And that, in fact, the statistician on a particular trial really helps define how those endpoints are met and how that all works. And I just, and I should have started out, you don't work on NET clinical trials only in the case of like the Vail workshop where you will get one. We had one on a NET drug this last time. You work with pediatric clinical trials, but the process is really interesting. And even inside of your world, there are difference of opinions on how to do trial design and stats. So I've done a long-winded introduction because I found the whole thing so fascinating. If you want to just give a little background of how you view clinical trials and your roles in it for patients, because I really think that you guys really do, and gals really do, direct the drugs that may come down the line for us in the future in ways that we don't understand and ways that might be interesting if you're participating in a clinical trial. So with that introduction, I only took 7 minutes to do that. I'll let you go.
Alrighty, I'm gonna talk a little bit about I was talking with somebody about this yesterday. My fundamental view is everybody gets treated. Patients always get treated, even if they aren't on a trial. And the goal, what separates a trial from just regular treatment is you're going to get data together and learn something that you can apply to everybody else. It is how can I get generalizable knowledge that I can use to make inferences so I can move the field forward. I understand that it's about treatment, but my job is to layer on top of it. How can we determine something that we can use to advance to the next stage of the clinical trial and to benefit patients in the long run? Everybody, not just the patients being treated. So the way I approach a clinical trial is I talk particularly with the clinical investigator and say, what do you want to learn from this trial? I'll ask them, is this the first time you've ever tried this therapy in a patient? If so, do you know if it's safe? And what do you mean by safe? Do you mean, can I give it for a year? Can I give it for just 28 weeks? So we go through on what people's definitions are about safety. What do you mean by efficacy? How do you want to measure whether the drug does a good thing? Curing people is everybody's ultimate goal. But for pediatrics in particular, that's something that happens maybe 40 years down the line. So we have to figure out good surrogates for telling us how drugs work, and we make sure that when somebody starts their trial, they're collecting all the necessary items to let them figure out, A, is the therapy tolerable by our definition of tolerable, how you help me decide what tolerable is, and is it efficacious? We also help investigators understand that many trials, you have to go in steps in trials. You don't ever try and go from my drug is the best thing since zippered pants, and in 2 years I will integrate it into primary therapy. There's very good logical ways that say I'm going to run a trial, I'm going to look at the data, and then I might decide we can't do it anymore, or there's potential. There's lots of new therapies coming up, and we have to get our clinical colleagues to think of the steps by which research proceeds, and doable chunks, if I can use that word, studies that advance, and good breakpoints to say, well, I can't give it feasibly, I guess we're done looking at this agent. Or I can give it feasibly, and I want to get a sense of how effective it is, and then I'll go on to the next step, knowing that at some point we want to get it into treatment, curative therapy for patients. So understanding, part of what I understand is what is a reasonable chunk, and try and think of a study that takes maybe no more than 4 years, because you have to have good decision points, right? We all make our decision points to yes and no decision points to go forward. It makes things doable. And the other thing that I have to remember is the FDA has put a lot of guidances about what are considered believable endpoints for tolerability, for efficacy. It's my job to understand what the approval process, what would support an approval. Like, don't invent a novel endpoint. There are many endpoints out there. They're very good. Some of them might not quite fit what you want, but I have to understand what they are, and I have to understand how they can be integrated in your trial, because we always have to have the view that at the end of the trial, we want to move it forward. And if we choose novel endpoints that nobody believes or have no grounding in the approval process, we've not done anybody any good. So I have to understand regulatory requirements to figure out what are endpoints. And another important job that I have is people always think they know how to collect data, and I am often reminded that common sense is not very common, right? That you have to have lots of experience, and what I try and do is take my experience and translate it into new trials to say, well, you shouldn't try and follow up on people every week. Try and follow up on them on every 28 days. Remember, have your evaluations correspond to when people are going to come in. Because if you make a trial hard to do for a patient, you aren't doing anybody any good, and you're actually only asking people to make your trial perhaps difficult, and non-compliance can really hurt a trial. So that is a very brief précis into how I see my job. Like, the main part of my job is in the design of the trial. People think that statisticians analyze. I don't analyze anymore. My graduate students analyze. The biggest effort in a trial is getting the design right and the data collection right from the start. And that I spend a very large amount of time doing that.
So, and I know you and I work in Vail on phase 1, phase 2 trials, and in my other, with my other hat, I work on phase 3 trials, and I'm often surprised that you can get something that's all the way along to a phase 3, and you get questions that with the support, with the data that you might get, you won't answer the questions. And I think what you're saying is really, you know, and certainly when we deal with the young scholars, and I like the way that you call the people we work with scholars at Vail, but you know, you ask a question or you look for your objective, your primary objective, and the number of times that the primary endpoints, the data you would collect for the primary endpoints won't support that primary objective. And that, you know, one of the things that I try to get through to the young scholars is that that doesn't help anyone. You used a bunch of resources, which are both dollar resources, but also patient resources, and you haven't answered the question. And that's not valuable. So the fact that as a statistician, you work in early to make sure that whatever those endpoints that are there will actually support the objective, which sometimes they don't even look like they'll align. I'm always surprised.
And the other thing that I try and impress on our scholars is that the worst case is not having an answer. Even if the answer isn't what you expected. A clinician that I work with a lot says, no matter what the result of the analysis is, that should always tell you something, and you should always know what the next step is, no matter what that answer is. So framing a good way to frame a trial is, what are the possible answers I could get, and where would I go from there? And one of my favorite expressions is, you've just seen your article in the New England Journal and you've opened it up, and what displays are you going to show, and what does that tell you about where you go next? Because we always have to think forward out of the trial.
Right. And I think the other thing that you brought up that was really interesting is that trying to answer too many questions in one trial leaves you with, well, one, not trials that are doable, because you may have too many things happening at one time and not being able to answer them. But that does, you know, impact, and one of the challenges obviously for patients is that it does impact the time that a promising drug or any drug will take to market. But that's what we need to do to find out if it's efficacious, as opposed to just randomly throwing drugs out back in the old, you know, Barney days, so to speak, or carnival days. The average time, I think, from a phase 1 trial to, if it gets through all those steps correctly, to a registered drug, I think is still 10 years. Is that about right?
That's about right. And, you know, it does take a while. We're working on people. The disease process takes a lot of time to evolve. People are biological units, and they react differently. And assembling those data is a difficult thing. There was another one that I haven't got to use yet. Is any investigator only has so many attention points to spend. Like, you don't have $1 million attention dollars. You only have so many attention points, and you have to think of how much you're going to spend on each one. And really, to do a good job, it does take a lot of work. It takes a lot of your time to understand it. And it's good to, I think, understand what people hope to accomplish and how they're going to accomplish it. I know that people think that they can do it on the weekend. You can't do it on the weekend. Some of it is just educating people how much real effort and a lot of it is building up strong personal interactions for the future to get people to cooperate. Because I think the one thing I found out about young investigators is they need some help at establishing relationships. They can build them with their colleagues, build them with their patients, because that's really what propels studies forward to a good conclusion.
So, and I know one of the other, I'm for lack of a better word, hot topic, but it's come up almost every year in 3 years, is dose escalation studies that, you know, we have some simplistic ideas of, and not to go too technical, because I promised people I wouldn't go too far in the weeds, but we still see things that are 3+3. And that's you try 3 people on a trial, and then if everyone goes okay, you go to the next highest dose. And do you want, I don't, I'm, you know, putting you on the spot, but quickly explain why that's a challenge and why we might be leaving good drugs behind or bad drugs forward.
So this, I think the 3+3 design that we're talking about, I traced back its origin not to any scholarly publication, but to a paragraph in an NCI investigator's handbook. And I couldn't figure out from there where it came. Somebody kind of wrote it out and said, I guess this is okay. There's been a lot of work on why this is a good thing. There are a lot, and the interesting thing about this design is it tends to settle on doses that are probably too low. That because what happens in this design is you start up, and the minute you run into trouble, you have to start to go down. I'll have to remember that flash that came up about the vaccine. That's a little different, but I do have an opinion. It goes down again. But and the problem is with 3 patients, you can just get bad luck just because you have 3 frail patients. Some of the more modern designs try and allow you to peak back up again to get a better sense of how the dose is related to the probability that you have a bad effect. So there are some pretty interesting new designs that try and estimate the shape of dose and toxicity, and provide, the other thing these new designs do is provide some early warning signs that you are at a dangerous level and have extra special stops for too much toxicity, or in particular, too much toxicity of a certain kind. When we write, can I go a little bit into the weeds in dose?
You can visit the blades of grass.
Okay, so when we, in a phase 1 trial, new therapy is given to a patient, and when we classify the bad effects that can happen, the side effects, by stuff called dose limiting, which are very serious side effects, and other things that happen that we tabulate just so that we can know about them. These dose limiting toxicities, they're measured on a scale that the NCI gave us, and they call grade 3 serious, grade 4 life threatening, and grade 5 death. And a lot of studies just say any grade 3 or 4, even though sometimes a grade 3 toxicity is not all that serious.
And it reverses itself. Right?
Right. Sorry. And what these new designs do is they allow you to identify toxicities of very particular kinds, like liver failure or something like that, that allow you to close down escalation if you start to see lots of these very concerning toxicities. These new designs are well thought out. The field is moving very rapidly, and we're trying to get our clinicians who work with old fogies like me to try and adopt newer methods. So these new methods, again, don't just get 3 and hope nothing bad happens. Try and figure out what the observed toxicity tells you. Are you in a safe area in and around that point, and should you go up? Or if you've gone down, maybe you should go up again. So these are called Bayesian designs. They're they're quite safe, and we are investigating more and more. And thankfully, we're moving away from this very old, as far as I could pin it down, 1956 technology for doing dose escalation. Just safer for everybody, patients, investigators, all around.
And I know one of the really big challenges, and certainly we see this, and I think, you know, we think of these things happening fast, but not all drugs do both efficacy and or toxicity show up quickly. And I think that is one of the challenges, because, you know, if you're doing a dose escalation trial, you can't wait 4 years to see if you have a dose limiting toxicity on your first cohort, or it would take next to, you know, infinity to get that thing done. And so how do you evaluate for longer term toxicities?
I'm glad you asked that, Josh, and you'll get your quarter later through the mail. We've, in particular, and this is a particular concern in pediatrics as well, we build into our trials after the first one, after this dose escalation scheme, every phase 2 trial I have has a monitoring rule that says, essentially, the dose I chose from this small phase 1 trial is too high, and I need to go back and recalibrate. And even my phase 3 trials, I'm working on a phase 3 trial now of a drug called cabozantinib in the space of osteosarcoma. And my trial now contains monitoring for 4 separate areas of concern: wound healing, long-term toxicity, that we've embedded in the phase 3 trial, because we acknowledge, as you said, Josh, the trials, these early trials, they get a signal, but the signal is crude, and a well-written trial always ensures patient safety, even at the phase 3 level. And again, I have 4 monitoring rules that I have to repeat every 3 months to make sure that any complications I'm seeing are within the realm of what would normally be expected for this very serious disease. So good trials never quit looking at safety.
Right. Interesting. And there's some of the things that come up. And funny, I was working on one of our trials actually last week. Post the workshop, I had set up some meetings, and now that's a different topic, which had some long-term toxicity, but not only that, longer term as to show efficacy longer than life expectancy, which we didn't know when we were in the workshop, but well, it's interesting, some feedback I got on trial design of make sure the patient will live longer than it takes for the trial to show any efficacy. Otherwise, that's, you know, when you and I were talking, or when you were talking to the scholars, one of the things is, look, once you enroll a patient into a trial, if something happens to that patient that has nothing to do to the trial, it still counts as a trial. I mean, as something on the trial. So if one of us enters into a clinical trial, and for whatever reason we get struck by lightning, hit by a muni bus, that counts in the trial and has to be explained. And that was, you know, as you're taking a look over the long term, right, you have to explain a lot of things that may have nothing to do with your trial, which I find fascinating and challenging, because you know, between the time a biopsy needs to be done and things, a lot can happen.
One of our reasons we do that is that, and it's, you know, it's as usual, we do things because somebody did something bad in the past, and now you have to figure out how to not do it again. There were people who were a long time ago who were running trials, and they were excusing bad events because they said, well, that had nothing to do with my drug or my therapy. And it was a personal value judgment. The data should be able to speak to you, and you should have enough knowledge of history to put it in the right context. So you should be comparing it with data that has those same other hazards and risks involved, so you can figure out a truth about what your therapy is what your therapy is doing. Right? Everybody counts in a trial. Everybody should count.
Everyone counts, regardless of whether it's, well, we don't know whether it was caused by the drug or not. So we count them and figure it out on the backside. One of the questions that come in is, you probably will speak to the averages rather than to your absolutes, but the percentage of clinical trials you've participated in or you've worked on that's led to an approval. It says approval of an effective drug, but I'm assuming that we're not going to approve any non-effective drugs.
But I must say, it's a little more difficult in pediatrics because by the time we get them, the only way they make it to pediatrics is they have to have some sort of approval in adults. There have been 4 that I have been on. I'm virtually so about half of the phase 2 trials on which I'm involved will promote the drug to a phase 3 setting. That means there is some good evidence of efficacy, maybe not cure, but effect on the disease. Only about 10% of them actually change fundamental therapy in the long run, because in children, therapies are pretty good, right? Leukemia, 95% patients are cured. I work in Ewing sarcoma. We're very upset that only 70% of patients are cured, and we're looking for a better treatment. In my phase 1 settings, about 3/4 of the drugs that have come through that have been targeted for children have eventually made it out of phase 1 and through phase 2 and then gone on to a phase 3 evaluation. Many of them in areas that I am not the statistician for. So I freed my children to do good things in the world. What eventually has happened, we do see a lot of success. And
so, but I will point out there have been studies on the FDA database, and the number is not all that impressive, especially in oncology. It's 3.4% of all drugs that enter phase 1 for oncology indication actually make it to approval. It's really small. Half of them, a little bit less than half of them drop out after phase 1. Only a third make it from phase 2 to 3, and only a third of those make it from phase 3 to approval. So it really is the funnel, and in oncology, the bar is higher, or not that the bar is higher, but the success rate is not as high as other disease indications. There's another question in here, and this one's interesting to me. And you brought up that you think down the line towards registration of a drug when you're looking at a trial. And this question is, if you could change one thing about how the FDA does clinical trials, what would it be? And I would say maybe we should rephrase that, because the FDA doesn't actually do clinical trials. The FDA you might have an advisory meeting about what they're willing to accept. But as far as I know, they don't do any clinical trials, I think. But if you could change one thing about the environment to which clinical trials are done towards registration, is there anything that is in your sweet spot of what should be done?
That's a tough one. I think, although people find, some people say the FDA is obstructive, I just don't want antifreeze in my aspirin. And I understand that they do a good job, and they do what in some ways is a very tedious job, but they do a lot to protect us. I'm not sure how I think things to me move slowly. I think a lot of it is just in this in the space that I've been in. A lot of the discussion, how we move studies forward, how the FDA responds to us with recommendations, now how we have to go back and redo our trials and hope we got it right. It's a very structured and formal interaction that, to me, during the design phase, slows things down a lot. And I think part of it is the regulations about how much interaction I can have, how much formal, how much I can take the FDA's recommendations, and now I have to go back and reintegrate them, and it goes to review again, and there's a lot of back and forth in that fashion. So that's where things have taken a lot of time for me. Now, getting the data together for a trial, when I present it for approval, I have found that I have found that very time consuming for me, but that's on the statistician side. On the consumer side, the data that I've submitted and I have watched the FDA go through, and I am impressed with how assiduous they are and how they make sure that everything I have done supports my claims as a statistician. So I don't have trouble once the study gets going and what FDA does to make sure that the data are right.
And I think you brought out a really good point there. One of the big differences between the US regulatory framework and, for instance, the European framework is that the FDA really does rerun the data as if de novo, as if it never had been seen before or run by a statistician to try to recreate the entire environment, as opposed to just accepting, in your case, your reports, but in most cases, a farmer report that may not have the same level of diligence and or be as conflict-free, so to speak. And I find that kind of interesting in the chain, the difference between the two, because twice I've been at on an advisory committee to their advisory panel at the FDA for drug approval. I'm just impressed about how much they've done to the data to look at it and scrutinize it in ways that even the provider may not have looked at.
And I understand that we could speed things up. As you said, when we presented to EMA for muramyl tripeptide, MTPPE, the data were not reanalyzed. They took our analysis and acted on it. And when we presented it to FDA, they actually reran it, and I had to answer questions about the data. We could speed it up, but I think then it's beyond my pay grade to say what risk are we willing to take on the public's behalf to say that if we're presented with data, we are going to believe it because no one will ever make a mistake, nor will they lie to us. So, it would speed it up, but I kind of understand why they are obliged to do it. And part of it is in the law, right? So I don't know. It's a discussion. It's an interesting discussion. If we move away from it, we have to understand fully the risks and benefits, which we are, because this evolved. We have to remember the history that got us here. We can't just say, oh, it's a bad place. A lot of reasons it's a good place.
No, I mean, it evolved because bad things happen. I mean, much like, when I was doing my research for giving the informed consent lecture this year, it's hard to talk about informed consent without talking about bad things, about places that didn't do informed consent or trials on humans that were done with, just really awful things, whether and it's not just, you think about just, World War II and things that happened badly there, but our own government did pretty bad things in order to get us to this place for informed consent as well.
Yes. Yes.
So there are reasons that rules are really there. So you've been gracious with your time, and I hope you were able to. You've got more things. One more thing. Go for it. You can have two more things.
Okay. This for me is can you help me out as an advocate? Or first off, we have an advocate committee in COG, but the thing that I really think we miss is an advocate on every study committee. I mean, somebody that understands the transactions of the study, because I find it, and this is because I'm on the outside for some things, when we have a list of objectives for a trial, I believe that I've made a contract with every patient that what they're doing answers a question, and that we have to answer every possible question. And when we don't do it, we are not doing right by those people who sacrifice their time and their resources to get it done. So it is always good to say, but what happened to secondary aim 2.3.1? Where is that represented to me? How can I find that out? Because I did have my blood drawn to see whether the trough level in cycle 5 was the same as the trough level in cycle 2. I do those analyses myself. I sometimes have trouble being on the inside for getting accounting of if we did this work and that it should pay off, right? Everybody should expect that what they did shows up somewhere in the literature. We want to advance. Somebody made the aim because they wanted to advance something.
Well, and someone agreed to a biopsy because they were willing to do it and put an extra risk. I mean, I think that is the, although I don't want to say bad things can happen in a blood draw, because there is that infinitesimal chance that it will, but adding more biopsies to a trial just because you need to have some reason for it and think about what you're going to report for it. And I think the entire faculty, oncology, patient advocates, and stats have been really firm about, if you are taking a biopsy, let's show us how that will count. Show us where you're going to use it in your stats. Show us where you're going to use it to report on. And if you can't do that, then take it out, because that's unfair to those of us who put ourselves at risk to do these things. Interestingly enough, thinking about putting at risk, I think the other thing I learned in clinical trials through this process at the Vail workshop, and you always bring it up, and some of the oncologists do, is all those aims, all those secondary endpoints that you might put in as a young investigator, when you put them in there and they show up in clinical trials, you're required to report. I mean, what's the reporting requirement and legally what you must do in order to, so someone doesn't come after you?
And it is clinicaltrials.gov. It's in the FDA, the FDA Amendments Act, this place where you deposit basic results on the trials, who got on, what the primary aims were, what the secondary aims were. Many investigators are very cognizant that exploratory aims are not reportable through clinicaltrial.gov, and they say, well, thank heavens, I don't have to report on it. I try, and every chance I get, for anybody else who can help me, make sure that, like, when you get we do now a lay summary of the results of every one of our trials that is posted and goes through our committee to summarize the results of the trial. At that point, I always encourage the people writing the lay summary, which is our advocates and nurses, to go back to the chair and say, but what about that aim? Where is that in the lay summary? To prompt people, to remind them you owe people, like, pay up now with a publication. I don't care if it doesn't get into New England Journal. Put it out there, because, you know, without the knowledge, you're just going to float around. Get that knowledge out there. So no box away.
Yeah, no, it is interesting, and seeing, you know, 23 secondary. I'm trying to think of what the record was this year for the largest number of secondary games in a trial. But there was one that was pretty big, which is okay. I mean, it's not okay. At the end of the day, you're not going to get it done, and you're going to leave institutions, and that's the challenge of doing that. Is there anything, before I stop the recording and stop streaming, is there anything you want to add to the patient community that you may not have hit? This has been just a wonderful discussion of why stats matter without having to, like, relive our statistics class. So let's do it this way. A lot of us are on drugs that have made it through the last, you know, that are approved. Is there anything we should be looking at when we look at the trial that got them done to understand? I try to make sure people understand that they need to look, that one, trials aren't cross-comparable because they have different inclusion criteria, and maybe even different, well, likely even different primary endpoints. Even if the endpoints sound the same, you have to look at how the endpoint is actually calculated. And so that's a blunt tool that says you can't cross compare, but also the way that the statistics done can be different between these trials. And is there anything from your neck of the woods that for patients that are looking at the, you know, whatever that New England Journal of Medicine article is, that we should be looking at? Or anything else you want to add to close out?
Anything, well, a good trial, something that you can, and I think this is both in when you read about it and when you participate in it. A good trial counts everybody, and a really good trial randomizes and counts everybody. I think there's a lot of hype about a drug that cured, that had a good response in these really late stage patients, and I am a big believer in randomization. I'm on at least one randomized trial myself, and I am all for it. I think it's the way, it is a commitment to accept the randomization, but I also think it's my personal contribution to how I'm going to make things better, that I'm not going to have it be just a folklore tale about how this group of patients did better, actually comparing things. One thing that we and pediatrics, were lucky, we have good follow-up on patients. We follow patients for 10 years, and our group is very committed to telling us what happened and staying in touch. And we have a long-term follow-up center that allows us to figure out what happened late. Like, we know now that people who are cured from childhood cancer really are at very high risk for many other bad things that happen afterwards. It's not like when you're cured, you go out into the world and you're like everybody else. Staying in touch is a good thing, because your continued effort helps people in the future. Because, as you said, Josh, many of these trials take a long time, and many adverse effects, and some good effects, take a long time to display themselves. Keep in touch, and let people know. We always want to hear about things. They help guide what our overall conclusions are. That's about the best I can think right now.
That's pretty good, Mark. I'm going to, let's see, stop the live stream.